This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 116. Recorded on November 19th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there.
Elio, from time to time, I pass a restaurant near my home. It's an Italian restaurant, and the name is Elio's Ristorante.
Ah, che bella cosa.
And I always want to take a picture of it to send you because it reminds me of you all the time. It must be good, right?
Of course. I'm sure it's delicious. Elio is not an uncommon name in Italy, by the way.
And it's good to have you back over Skype, having seen you in person for our last episode.
Yep. It was fun.
Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
Welcome back to the Skypes.
Yeah, it's been a while since I strapped on the headphones.
Yeah, are they working?
They're working great, I think.
I think one time they were broken. You got a new set or something?
No, I used some electrical tape.
So utilitarian, handy.
Resourceful.
All right, and Michael Schmidt is in Oregon today, so he can't join us. But we will have your ears and tell you about some cool stories in microbiology. This episode of TWiM is sponsored by ASM Biodefense and Emerging Diseases Research Meeting. Early bird registration rates for the 2016 ASM Biodefense and Emerging Diseases Research Meeting are ending on January 14th. If you want to go to this meeting, register now and save some money. The meeting is at February 8th through 10th in Arlington, Virginia, the premier event to discuss how to defend against the growing threat of bioterrorism and shape the future defense research agenda. And I must tell you that the last 2 years I have gone to this meeting. We've done a podcast, we've done a TWiV and a TWiM, and we're going to be there in 2016. So I will be doing a This Week in Microbiology from ASM Biodefense and Emerging Diseases Research Meeting. So come to the meeting and say hello. Be happy to meet you. You can learn more about this meeting at asm.org/biodefense 2016. That's all one word, biodefense 2016. We are going to start with a snippet from Elio.
Okay. So here's the snippet.
It's a great name, by the way, which we have taken for TWiV as well, which you originated.
Well,
snippet. You invented the word snippet.
Whatever. Anyhow, the story. Well, first of all, let me ask you a question. Mm-hmm. Who invented agriculture? You're going to say the Sumerians or the Assyrians or somebody like that in the Middle East?
Or could it have been microbes?
That's it. It was invented by insects, actually. Not quite microbes, but they used, what they did is they invented agriculture in its full splendor. And the best example, which I think many readers have seen on PBS or something, there's usually a beautiful movie that can be seen showing ants cultivating fungi. These are the leaf-cutting ants that exist in tropical America. And if you go there, if you go to south of Texas, actually including Texas, but many south of Texas, you find that these guys come out of huge underground nests. I mean, these are nests that are 20 feet across. And they go and collect and cut up a piece of a leaf that's about 3 times their size in length, and then carry it just like a person would carry an 8x4 board back to the nest. And do they eat the leaves? No. They chomp away at them and triturate them. They reduce them to little particles. And this chewate, as I'm fond of calling it, becomes what they inoculate with fungi, which are there in the nest in what are known as fungal gardens. And when the fungi grow, they eat the hyphae, and that's their food. And it's agriculture at its best because they prepare the ground, they prepare the soil, they fertilize it because they do that with the feces and the saliva. They seed it, they crop it because if the fungi get too big, they can't eat them. They become mushrooms, actually. They stick out of they get to be huge. So they have to crop them and then they have to harvest them. So they do everything that a good farmer would do. And this was invented 40 million years ago, at least. Turns out that termites do something very similar. Termites actually don't eat the fungus so much as they eat the fungus breaks down the woody material that they bring to their nest. Some from your house. You ever had problems with termites?
Yeah.
Either of you?
Oh yeah.
Not that I know of.
Okay. Well,
that's the worst
they're
kind.
Tough. They're tough. Anyhow, so we add to that, and there are other insects that do this, like certain beetles and so forth. But a new one is that bees do that. Now, bees are not known to do anything of the sort, but a group of Brazilian investigators, the name are Menezes, Voledneto, Marsaioli, Zampieri, Cardoso Fontura, Ducati Lucchese, Imperatriz Fonseca, and they're from several Brazilian institutions. This is a paper that is published in Current Biology, November, entitled "A Brazilian Social Bee Must Cultivate Fungus to Survive." And this is news. So what they do, and what's news is also how they use this fungus. So let me tell you about these bees. These are social bees. In other words, they have hives, they make honey, and they're interesting because they're stingless. So you would think that, I don't know if the production of honey is as good as that of the stingy ones that we have here, but you would think that there's probably a reason for it'd be nice to use them for making honey. But anyhow, they are used in Brazil for that purpose. So the bees make a hive. The hive is made out of a thing called cerumen. And cerumen is what we call wax. You know, we think of beehives being made of wax. Well, they're made of a combination of wax and plant resins, and it's known as cerumen. And they make these beautiful hexagonal shapes, and in each one, they place an egg. The queen, which is, in the social insects, usually the only productive one is the queen. So the queen lays an egg, and then they seal the chamber, wait for the egg to develop. But the egg would hatch and have nothing to eat. So what do? They regurgitate some food and place some food in this chamber so that when the egg hatches, there is something for it to eat. Okay, that is okay. But they noticed that when they opened up prematurely some of these cells from the beehive, they noticed there was a fungus growing, a mold growing in them. And they wondered, what's that all about? And they waited for a while, and then they saw that the mold disappears. So what did they conclude? They concluded that the larvae ate the fungus. And there's a beautiful movie in here. In the show notes, you'll probably have a link to that. There's a beautiful movie that shows how these larvae are chomping away happily at the fungus. So that's great. But there's more to it. First of all, you have to ask the question, why do they eat the fungus when they are provided with food, right?
Mm-hmm.
Workers, before they sealed up the chamber, have regurgitated plenty of food. That should be enough. Well, it's not quite clear what they get out of eating the fungus, but what is clear is something different. If you do not have fungi in there, which you can do by sterilizing the food with UV, the larvae die. And why would they die? Because without the fungus, the food gets contaminated and smells bad, and the larvae can't eat it. So that's the story, that the fungi somehow keep the food from spoiling. So besides eating the fungus, they must eat this other food as well, the food that's provided to them. It's not quite clear what goes on here. It's not known what food is gotten from the regurgitate and what food is gotten from the fungus by eating the fungus. But what is clear is there's no fungus, no bees. So this is essential for the life of the bees. And that's sort of pretty much the story. I'm sure that they're going to investigate what components of the fungus are available. The fungi, by the way, are kind of interesting. They are ascomycetes molds from a genus called Monascus. Monascus is, and they did some genetics on it, or some genomics on it. It's related to something Monascus ruber and Monascus pilosus, which I don't know. But Monascus is used in Asia to make a thing called red rice. And red rice is simply rice on which this Monascus pigmented mold grows, and it gives it a certain flavor, which apparently is prized. So Monascus actually is known for making a huge number of secondary metabolites, that is, products whose function for the fungus is unknown, but which are bioactive in various ways. So anyhow, that's an interesting combination. So there is a symbiosis here going on between the bees, the fungus, and who knows what else.
So this is probably something secreted by the fungus?
They don't know. They don't. I mean, they eat the fungus, So yeah, I mean, the fungus has to do something. It has to create something to keep the original food from spoiling.
Right.
I think this is an early paper, so we're really, we should wait and see. By the way, the red rice is traditional not only in Asia but also in West Africa, where it's called jollof rice.
There's been a lot in the papers about the collapse of bees.
Mm-hmm.
Do you think use of insecticides or antifungals?
Well, certainly using fungicides, and if this is true for the northern bees that we have, then it's bad. It would be very bad. Obviously, you don't want to mess around with this kind of symbiosis.
Yeah, an unintended consequence.
That's right.
So Michele, you're thinking that the decline might be due to reduction of the fungus.
I'm wondering if that's contributing.
Yeah, it could. It's probably multifactorial.
There are many theories, aren't there, about the colony collapse syndrome, it's called.
Yeah.
They have mites supposedly involved. They have who knows what else. But fungicides would do it.
Yeah, I don't think anyone has looked at this fungus and whether it is ubiquitous in all bees. Do we know? Is this the case for all bees, or is it just this certain species of bee?
I don't know. I really don't know. I couldn't tell you. I don't know.
It's a very interesting idea, Michele. I like that. This is great. This is a great
it's lovely biology.
Association. Yeah, I'm really happy that people can do this kind of research. You can imagine it would be hard to support unless the Honeybee Association supports you, right?
Oh, by the way, I should have said that leaf-cutting ants and wasps make antifungal products themselves because they are very picky about which fungi they eat on their regurgitate, on the chewate.
Those are technical terms.
It's only certain fungi that work. So they make an antifungal antibiotics which inhibit the growth of the unwanted fungi but permit the growth of the wanted one.
All right.
How about that?
Yeah, that's pretty cool.
Yeah, that's really neat.
This is a nice story. Our friends in Brazil Elio, they have names that sound Italian, right?
Yeah, well, a lot of Italians settle in Brazil and in Argentina.
Yeah, yep. Yeah, I have some relatives down in Brazil.
Right.
All right, thank you, Elio. Of
course.
All right, I have a paper for everyone, which is in a brand new journal called M-Sphere. Let me just take a few minutes to tell you about this because it is an open access journal. You can actually get the papers that we talk about. Open access. And it's published by the American Society for Microbiology, for whom we have a lot of love. This is a wonderful society, by the way. of many, many microbiologists. It is a society to promote the microbial sciences. And even if you're not a microbiologist, you can join and take advantage of many of the things it provides. And there's some very cheap membership rates as well. So you should check it out and support microbiology. So they publish a lot of journals. They've just started 2 new ones, and this one is M-Sphere, an open access multidisciplinary journal that is going to broadly publish within the microbial sciences. They will publish articles on viruses, bacteria, parasites, fungi, broad areas as well. And so if you work in any of those, you should think about publishing. Last week, I think they published their first articles.
So it must be a very smart scientist who's decided to be the editor-in-chief.
I was just going to say, and I know how to pronounce his name now. Yes. Michael Imperiale is the editor-in-chief.
And he works where?
Yeah. He works at the University of Michigan in Ann Arbor.
Yes, he does.
Not too far from Michele Swanson, right?
Yeah, and he had served for a number of years as editor of Journal of Virology and MBio, 2 other ASM journals.
That's right.
So it's great that he's got the opportunity to lead this new effort.
And I should say something that bears saying. Right now, we are suffering from a plethora, an avalanche, a torrent of new journals. A day doesn't happen that there isn't a new journal. I mean, PLOS has exploded. And then actually my email inbox is full of emails that say, would you like to contribute or would you like to be an editor of our new journal, from all over the place. And it's a disaster. However, it is nice that in this crazy world, something rational is being done. And M-Sphere looks so totally rational, and that I think it will have a future.
I agree. I think it now more than ever, it's really valuable to have a professional society backing a journal, because then you know there's quality control, and it's going to be excellent.
So, there are lots of new open access journals, but only a few are really the real deal. The PLOS journals are very good. ASM journal, well, MBio was the ASM's first open access journal, and it's done very well.
Oh, that's a dynamite journal. My God, that's really way up there.
The editor-in-chief, Arturo Casadeval. Now, I mean, you have to figure out how to support an open access journal. That's the key. So you have high quality material and have it open access, which means that the readers don't have to pay. So you have to have some other way of supporting it. And that's really the key. And so I have high hopes for M-Sphere. I'm sure Michael Imperiale will do a great job. He has a host of senior editors in all these different fields to help pick papers. And then there are editors below them. And I have to say, in full disclosure, I am an editor of M-Sphere.
Well, thank you for your service.
Aha. Now it comes out.
I don't get paid, believe me. None of us get paid. We do this for love of microbes. Michele.
I'll also add that I spoke with the first author, and she said they had just a great experience with the journal. It went very quickly. They got terrific reviews that were thoughtful. So she highly recommended people submit their work there.
By the way, Michele, your mic seems to be doing funny things.
Oh.
Yeah, that's perfect.
Is that better? Okay.
You had it away from you or something? It's good. That's perfect. Don't move it. In fact, don't move at all, Michele. Yeah, so I had a little conversation with Michael Imperiale and Pat Schloss, who had the first paper accepted at M-Sphere while I was in Michigan, and hopefully that'll come out as a promotional video at some time. But as an editor, by the way, we had this unusual idea to make me an editor so that, I have podcasts in virology, microbiology, and parasites. And so we could actually look at papers in M-Sphere for all of these podcasts. So that's the plan. We'll do a paper now and then on TWiM, on TWiV, and TWiP. And then the journal will link to our podcasts. And from time to time, we will actually have the authors come on our show and spend 15 or 20 minutes, right?
That'll be great for teaching, for undergrad and graduate courses.
Yeah. So, in fact, on TWiM, we have a paper by Carolyn Coyne coming up on M-Sphere. We're going to have her join us, and the student, the first author on that paper. So we're going to try and facilitate that. So when you hear in TWiM in the future people coming on, that's why.
So Vincent, when you say editor, what do you mean?
The little I stands for internet.
And how is that different from...
Oh, I get to pick papers for the podcast and we discuss them. It basically leverages our podcast audience to...
So in other words, you're not an editor for the print.
Well, and technically I could handle papers as well, right? But my primary job is to look at them. Every week I get a list of accepted papers, and sometimes the editors will say, this is cool, you should look at this. And so today's was an example of that. And then
you get to focus entirely on bringing it to the podcast, et cetera.
Exactly.
Great. That's great.
So I want to use social media to help launch this journal, which I think is different and really hasn't been done before. So I'm happy that ASM went for that, and Michael Imperiale. And so today we have our first paper in M-Sphere that we're going to talk about, which is a very nice paper. And the editor who handled it was Aaron Mitchell, who used to be my colleague here at Columbia. He works on Candida, a kind of fungus or yeast. And the paper is on that today. And he has since moved to Carnegie Mellon, and he is one of the senior editors of M-Sphere. And the paper he recommended we talk about, among the first to be published in M-Sphere, it's called Manipulation of Host Diet to Reduce Gastrointestinal Colonization by the Opportunistic Pathogen Candida albicans. And the first author is Carney Gonzalez, and followed by Tornberg, Belanger, Mathan, Lichtenstein, and Kumamoto. Carol Kumamoto. These individuals are from Tufts University and Tufts University School of Medicine, where Elio used to be, right?
That's right.
And you knew Carol.
I know Carol very well.
And that's when I first met both Elio and Carol.
Really, at Tufts?
I was a postdoc there, yeah.
With Ralph Isberg?
It's a great department.
Yeah.
Yep.
Elio made a good contribution to that department. He really built it up.
Oh, I didn't screw it up. Let's put it that way.
Negative results. Negative results are good. It's okay. Now, it so happens, Candida albicans, I sort of know a little bit about because I had to study it a couple of months ago to make these lectures that I did in Germany. And this was one of the lectures I gave on Candida. And so it is a fungus that colonizes the skin and mucous membranes of most healthy humans. It grows as a yeast form, single cells, and it can become filamentous. And the title of this paper includes the word opportunistic pathogen. And that's because it causes disease in certain kinds of people, and it's opportunistic. So it's there as a normal commensal. In some conditions, it causes disease. For example, if you develop AIDS and you become immunosuppressed, you are likely to develop a condition called thrush, which is the growth of Candida in your mouth, and you have this white fuzzy coating on your tongue and
like the bird thrush.
Thrush, that's right. Lots of babies get thrush when they're first born because they don't have a terrific immune system. They often get it in their mouth.
Or on their head, it's called cradle cap.
Cradle cap.
Yep.
Wow. I didn't know that one. Yeah, I remember our first son had thrush. We had to treat him to get rid of it. So that's a superficial mucosal candidiasis. Or, if more seriously, the yeast can enter the blood and spread systemically, and it can cause invasive disease. It can get into the kidneys, liver, spleen, lungs, brain, heart valves. And this is associated with a very high mortality.
And right now, it's the fourth leading cause of bloodstream infections.
Fourth leading cause.
No kidding.
Yep. Other risk factors, by the way, besides being immunosuppressed, if you get broad spectrum antibiotics for surgery that you're going to undergo or for some infection, it will alter your flora and allows Candida to overgrow. If you have a central venous catheter, it's a catheter put into one of the large veins in your chest to deliver something, this is a risk factor because the yeast can get inside of you. Now, so for these people who are at risk, we would like to have ways to reduce the risk for Candida infections. Now, you can use antifungals. There are a number that we can use for Candida, like fluconazole is a well-known one. But of course, if you overuse any kind of antimicrobial, you're going to get resistance. So in this paper, what they're trying to do is find a dietary way to reduce colonization, specifically in the GI tract, with Candida albicans. And we know from many discussions on TWiM and many papers that you can change your diet and alter your gastrointestinal microbiome. So if you switch to a high-fat diet, for example, can alter your microbiome. So in this paper, what they have done is to ask whether coconut oil can do anything to the colonization of the gut with Candida albicans. Coconut oil is a natural product, comes from coconuts, right? Has a known fatty acid composition, and it's rich in medium chain length fatty acids. All right. Now, fatty acids, to remind you, these are carboxylic acids. They have a COOH group at one end and a long tail of linked carbons with hydrogens. And these can be unsaturated, which means they have some double bonds, or fully saturated, no double bonds. They're all full of hydrogens. You can't put any more hydrogens in. And you may have heard people arguing whether saturated or unsaturated fats are good or bad for you. That's a lot of controversy there. But coconut oil has lots of medium chain fatty acids. And they compare this to beef tallow, which is beef fat basically, and soybean oil. And these have long chain unsaturated fatty acids. The coconut oil has mostly saturated medium chain length fatty acids. So they have a mouse model where they can feed C. albicans cells to the mice orally, and they basically give them a high-fat diet with coconut oil or beef tallow or soybean oil or a standard diet, and then they orally inoculate them with the yeast, and then they measure. Yes, sir.
Let me interrupt. It's interesting to figure out how did they come to the idea of using coconut oil? Well, there's a paper in a journal called Journal of Medicinal Food out of Nigeria called In Vitro Antimicrobial Properties of Coconut Oil on Candida species in Ibadan, Nigeria. And there's a previous paper to that also. So it was known that coconut oil and Candida don't mix too well.
Right. It's known to be fungicidal or fungistatic, right?
Probably fungistatic.
In vitro.
In vitro, right. It hasn't been looked at in animals, right?
Yeah. So they picked up on that idea. And I think one reason they would have known that is because some of the co-authors are not in our old department, Michele's and mine, but are in the USDA Human Nutrition Center at Tufts.
And you might wonder how the microbiologist and the nutritional cardiovascular expert got together. And Carney told me that they were actually Carol Kumamoto and Alice Lichtenstein were in line together to get their flu shots. And they were friends and colleagues, and they started chatting about what was going on. And through that conversation, this idea was born to look at the impact of short and medium and long chain fatty acids on the fungal population. So isn't that a great story?
It's another reason to get a flu shot, right?
Yeah, and talk to people, not stare at your phone all the time.
Yeah, you should talk to people. It's a lot of fun when you talk to new people or old people all the time. Yeah. All right. So they feed the mice one of these coconut oil, beef tallow, or soybean oil. Then they inoculate them with Candida orally. Then they measure colonization with the Candida in fecal pellets. They do time courses, or after 21 days, they sacrifice the mice. They remove the stomach and intestines, and you can take the contents and measure the amount of Candida. You can do colonies or PCR. Now, the coconut oil is given at between 18 or 30% of the food weight. So, they have a standard diet and they supplement it with this. So that's quite a bit. And we'll get back to that later. Now, one thing I want to point out, before this whole experiment, before it starts, they give the mice antibiotics to reduce the GI load. And they continue those during the experiment. These are to reduce the bacterial loads, I presume, in the GI tract.
Yeah, the Candida won't colonize unless you knock back the normal microbiota.
Right, so you can't do the experiment. I just wonder if, without that, so eventually this is going to go into people, and you're not going to be giving them antibiotics all the time. So I just wonder whether that'll impact that, but we'll see. All right, so if you feed mice coconut oil, the colonization of Candida in the stomach is lower than in mice fed the other things. Same thing with the cecum and fecal pellets. You find fewer Candida albicans cells. All right, so if you feed them the coconut oil, then introduce the Candida, you get less colonization. Now the question is, can you reverse Candida that's already colonized the gut? All right, so they give you a diet for 14 days of beef tallow, which gives you good Candida colonization. Right, beef tallow has these long chain fatty acids, and apparently with that the Candida go up a lot in the gut. And then they switch to coconut oil. So after you've established Candida, you switch the diet, and that decreases the colonization to be the same as basically mice given coconut oil throughout.
That was such a great result.
Yeah, so that's really good because in people, of course, you're gonna have Candida already there, and you want to know if it gets rid of it. So that's a good finding. The other cool thing is, if you feed mice coconut oil plus beef tallow, you still reduce colonization. So coconut oil can actively inhibit the increase caused by beef tallow. So if you have a high-fat diet, you could, in theory, take coconut oil if this works in people.
Yeah, have a steak and then a little bit of coconut something for dessert.
Have some coconut oil. Coconut oil is good, right? I like coconuts, so you don't often get them. That's the problem.
I'm actually going to share a recipe at the end of the show.
Very good. For something with coconut oil or with coconuts?
Coconut oil.
Excellent. So now we ask, what's it do? What is the coconut oil doing? They ask, how does it alter the fatty acid composition? So they sacrifice mice on the different diets. They take the gut contents and they do gas chromatography, and they look at the fatty acids. Okay. Remember, fatty acids are the components of triglycerides. So, and the fatty acids, I told you the characteristics of the three diets. So the findings with beef tallow, the GI contents have mostly long-chain fatty acids, as you would expect. Coconut oil, rich in medium-chain fatty acids. Okay, so this is exactly as you would expect, because the diets, what's in the gut is reflecting what they're eating. Now, interesting side note here, as you move from the stomach to the distal small intestine, the amount of fatty acid per milligram of the contents of the GI tract goes down, and this is something we knew before as these compounds are absorbed or metabolized. All right. How about if you compare the diets and look at the concentration of fatty acids? Is that different? We already looked at the kinds of fatty acids. Does the concentration matter? The concentration of octadecanoic and hexadecanoic, those are long-chain fatty acids. They're lower in the cecum of mice fed coconut oil or coconut oil plus beef tallow than tallow alone. So they're thinking maybe this has some contribution to the reduced colonization. You're getting reduced long-chain fatty acids. So now we have to talk a little bit about...
By the way, all these results are compared to a standard diet, which I guess has no added fatty acids.
That's right.
And it does make a difference with no matter which oil you add or tallow, the amount of fatty acids in the intestine is going to go up.
That's right. That's right. I have not mentioned that in the interest of simplicity, but that's absolutely true. And you can go read it for yourself. It's open access. Did I already say that? Yeah,
I sure did.
All right, let's do a little lipid metabolism. And don't go to sleep. This is really cool. I used to not like this in college, but...
Plus, there's a nice figure. You can follow along. They've laid it out schematically beautifully.
Yes, it is really nicely drawn. When I was in high school and I learned metabolism, intermediary metabolism, it was boring because they didn't put it in any context. Now we put it in the context of fungi. It really is interesting. So here's this, to take a quote from Elio, here's the story. Fungi can use lipids as a sole carbon source to make energy and the biosynthetic precursors that they need to build things. So they can use lipids. We can't do that. The reduced levels of octa and hexadecanoic fatty acids that they found in the previous experiment would suggest that there's less fatty acid catabolism in Candida, and that's possibly why it's growing more slowly. So they measure expression of the genes involved in this pathway in the Candida in the cecum of mice. So they take out the Candida, they do RT, reverse transcriptase, quantitative PCR, and measure these genes. And of course, they compare the different diets. Now, fatty acids are broken down to acetyl coenzyme A by what's called beta oxidation. You guys remember acetyl-CoA? It's a high-energy compound that's fed into the Krebs cycle, also known as the tricarboxylic acid cycle. And that cycle makes energy in the form of ATP, among other things. So fatty acids are broken down to acetyl-CoA, and the genes involved in beta oxidation are lower in Candida that are fed coconut oil or coconut oil plus tallow compared with beef tallow or the standard diet. So the implication is they're probably making less acetyl-CoA. And again, you need that to make energy. So that's pretty cool. I like that. Now, beta oxidation. conversion of fatty acids to acetyl-CoA happens in a structure called the peroxisome in yeasts like Candida. However, when they make acetyl-CoA, it has to get to the mitochondrion, but it can't cross membranes. So it has to be converted to a compound called acetylcarnitine by enzymes called carnitine acetyltransferases. It can then enter the mitochondrion, and then it's converted back to acetyl-CoA. It seems a little circuitous, but that's what membranes do to you. and the net gain of having a membrane is good, so it's worth the energy. They look at the expression of these genes encoding carnitine acetyltransferases, and as you might guess, it's lower in Candida from mice fed coconut oil or coconut plus beef tallow compared to the other conditions. So there's probably less acetyl-carnitine transport into the mitochondrion, hence less acetyl-CoA to go into the Krebs cycle. A little bit more of metabolism. It's okay, just a little more. In the mitochondrion, as I've already mentioned, you take acetyl-CoA, you make ATP if you go through the Krebs cycle, or you could also make sugars from it. That's called the glyoxylate cycle. So they looked at the expression of glyoxylate cycle genes in Candida from mice that got these various diets. And again, these genes are lower, their expression is lower in mice given the coconut oil or coconut oil plus beef tallow than in mice fed tallow or nothing except standard food. So glyoxylate cycle, that is the synthesis of sugars from acetyl-CoA, a breakdown product of fatty acids, which is what we're feeding the mice, is reduced by coconut oil. Now, some of the products of the glyoxylate cycle is glucose, and this part is called gluconeogenesis. You may remember that from high school, and you probably never wanted to hear it again, but in this context, it's very cool. So based on what we've learned so far, you'd predict that Candida albicans from coconut oil-fed mice would have lower gluconeogenic gene levels expression. It's because they have less substrate for gluconeogenesis, and the substrate is acetyl-CoA, and they would have higher glycolytic gene expression, breaking down sugars. So in the latter, glycolysis or glycolytic activity is a Candida gene called CDC19. It converts phosphoenol pyruvate to citrate, which can enter the Krebs cycle. And this is exactly what they found. CDC19 is higher in Candida albicans from mice fed coconut oil or coconut oil plus tallow than tallow alone. And one more metabolic note here. So this is very cool. They start out with the observation, and they dig deeper and deeper to try and figure out how it's working.
Yeah, they get into the biochemistry and then the genetics, the molecular mechanism. It's really great.
This is why we call it microbial sciences, because it's a whole thing. The expression of genes that break down fatty acids was also lower in Candida from mice fed coconut oil or coconut plus beef tallow compared with the other diets. So the implication of that is that Candida can use long-chain fatty acids from tallow as a carbon source, but not when the diet contains coconut oil. And the authors think that the absence of long-chain fatty acids, and these are fueling the growth of mice fed beef tallow, the absence of those in coconut oil-fed mice decreases colonization. So it's basically metabolic programming by dietary intervention. That's the way the authors put it. I think that's a really nice way of saying that. You alter the ability of the Candida to grow by changing the fatty acids that it normally would like to have to colonize. So basically, the bottom line is adding coconut oil to a fatty diet could reduce Candida colonization. And as Elio said earlier, coconut oil is also antifungal and probably contributes to reduced colonization. And the medium chain fatty acids are likely to do that. Now, again, these medium chain fatty acids are saturated. They have no double bonds. Their long carbon chains are all full of hydrogens. This has been suggested to be bad to have a lot of saturated fatty acids. It's a controversial thing. It's been associated with cardiovascular problems. So we don't know what's going to happen, but the next step, of course.
Yes. I thought in their discussion, they it was admirable that they tried to take us into the metabolism of the medium chain versus the long chain, and point out that they have different solubilities. They're actually processed differently. They traffic differently. One through the blood, the long chain fatty acids through the blood, the other through the lymphs. And so we don't have any evidence that these medium chain fatty acids would be a safety risk for your cardiovascular system.
Because the dietary saturated fatty acids that are supposed to be problematic are long chain, right?
Right.
But also when you think about applications of this idea, the place where you would use it is when you have cleaned up people's guts with antibiotics for whatever reason. And for a relatively short period of time, it may be days or weeks, you're at risk of colonization by Candida. But so you would only have to inhibit that over a period of a week or two, and then you reestablish the normal microbiome, and the Candida goes to sleep. So it's not a chronic. I don't see you in terms of human intervention that you would use this over a long period of time.
Right, I agree with that. Yeah.
The other population that they're actually now have IRB approval to test these ideas in are premature infants. So they've been able to recruit a collaborator from the hospital systems at Brown, and they have designed a study where they take premature infants who would be fed their normal diet. They could collect their feces and do the profiling and see who's there and how much Candida is there, and then introduce infant formula that already has got coconut oil in it. That, like for decades, that's been a component of infant formula. So they can add that naturally and then again collect the stool samples and see if there's been an impact in that human model. So that's really pretty exciting.
So that's going forward.
They have approval and they have the design. They're waiting for the funding.
Nice.
Yeah.
That's really nice. So I was going to point out that in an adult human anyway, how much coconut oil would you have to take? They say about 3.5 tablespoons a day. That's not so bad, right?
No.
In the old days, people used to make their kids eat cod liver oil, right? A tablespoon a day, and they hated it. But nowadays, they could make it. Coconut oil probably tastes fine, right?
Well, and maybe I'll jump in with the recipe and then back up and tell you about who shared the recipe with me. So the first author, the postdoc, Carney Gonzalez, we had just a great conversation yesterday, and she now makes her own, what is available in grocery stores is known as magic shell. So it's a combination of chocolate and coconut oil that at room temperature is liquid, and then when you squeeze it onto your ice cream, it immediately hardens.
Oh.
So she takes 1 part coconut oil, like a 1/4 cup of coconut oil, and mixes it with a cup of chocolate chips, for example, melts it and mixes it up in the microwave, and then stores it at room temp. And you can have it with bananas, ice cream, pretzels, your vehicle of choice.
So does she do this because of this study of the coconut oil?
She does it in part because it tastes good, but I think, she's a believer in this study as well. But as you pointed out at the top of the show, Candida albicans does colonize many people without a problem. It's just if you're on it, high antibiotics, or you have some other vulnerability, you're more at risk for disease.
Yeah. Well, it's good to be prepared. You never know when you need emergency surgery and they give you a lot of antibiotics. Might as well have that coconut store.
And why not add a little red wine to our diet and then also and the meal with some magic shell
for all diseases.
Good. Coconuts, red wine, anything else? Then you can eat whatever you want, right?
Well, that would be the ideal. Let's get those microbiologists figuring it out for us.
Well, next time we visit you, Michele, you can make this for us, okay?
I'll do that. Well, I think all of our listeners can make it now. But let me tell you a bit more about Carney. So she grew up in a house where her mom was had a law degree and was working in the ethics unit at the University of Illinois, Chicago, or Champaign-Urbana. So that's where Carney grew up. And her mom, because of her interest in ethics, professional interest, she had issues of science and nature laying around the house. So at a young age, Carney also was aware that she and her dad have really fair skin, and so they had a lot of discussions about skin cancer. So Carney would scan through the Science and Nature magazines trying to learn what she could about skin cancer. So she had an interest in science at an early age. She was in a math club in junior high, and a professor from the University of Illinois, Michael Plua, came and gave a talk to her math club and then invited the students to come to his lab for a lab tour. And according to her mom, she could not stop talking about this experience and how cool it was to be in the lab. So her mom said, well, just contact this person, ask if you can come in and, be a dishwasher. So she did that, and Michael said, well, I've got a dishwasher, but why don't you come in and start doing some experiments. So he would sit her at the blackboard, describe, what they were doing, and then teach her how to pipette. And so as a high schooler, she was in the lab doing experiments. So she said that was just a wonderful experience, and she, to her credit, is interested in doing the same, providing the same opportunity for others. So in addition to her research, she's able, through the Teaching in Education and Critical Research Skills training grant that's through the NIH that Tufts has, she and a group of about a dozen other postdocs spend 75% of their time on research, and then another 25% is protected to develop teaching skills and actually do some teaching. So she was able to teach a genetics course, a summer course at UMass Boston, which has a large percentage of underrepresented minorities. So she was able to contribute that important service as well. So that is her story. She also wanted to credit her colleague, Narupa, who did all the mass spec that provided, the biochemical cues to how this was working at a mechanistic level. So she had nothing but great things to say about the team they put together there at Tufts, and really wanted to give a shout out to Michael Pluva, who recently retired. He's emeritus, and he retired after receiving 34 consecutive teaching awards, 34 years running teaching awards there, and had more than 30 years of NIH funding. So she really just felt that was a great privilege to be exposed to his joy of science early on.
Yeah, Michele,
let me guess, is Carney still a graduate student or a postdoc?
She's a PhD student.
Okay.
Or, I'm sorry, she's a postdoc. She did her graduate work at Madison, University of Wisconsin-Madison, and was studying breast cancer.
Wonderful.
And then made the switch to this project. But she points out she's always been interested in how cells detect signals and then make transcriptional changes to alter the cell's behavior. So she did that in a neuroscience department studying breast cancer, and then is asking somewhat similar questions here now with Candida.
So, Michele, before we started recording, you taught us how to pronounce Carney's first name, but I have to say, here in New Jersey, we have a town called Kearny, and we pronounce it Carney. So I knew how to do that. It's right next to Newark, New Jersey. You know where Newark is, Michele?
Of course I do. I lived in Manhattan for 4 years, don't forget.
Yeah, I do. I'm just playing on your Ann Arbor place. But yeah, Kearny, New Jersey. Great name. Well, thank you. This episode is also sponsored by the 32nd Clinical Virology Symposium, which is taking place from May 19th through the 22nd in Daytona Beach, Florida. Wouldn't you like to go there? Abstract submission is now open. Submit your abstract before March 17th to debut your latest research on rapid viral diagnosis, the clinical course of viral infections, and preventative and therapeutic modalities for virus infections. You can present your work to over 1,000 researchers and primary care physicians at this important clinical virology meeting. For more information, go to asm.org/CVS2016. asm.org/CVS2016. Let's read a couple of email before we wrap it up. First one is from David, who writes, Dear TWiMeriti, thank you for your lovely show. I started with TWiV after taking Dr. Racaniello's online virology course and have since moved to TWiP and TWiM, all very instructive and pleasant. Yesterday, my daily Twix dose consisted of TWiM-19. During that episode, there was some speculation whether syphilis had been brought to the Americas by Columbus after having surged recently in Europe, or whether he brought it back. I am passionate about history, and the conquest of the new continent has been a rather merciless and bloody one, especially in the 16th century. Viruses and bacteria certainly played a major role in the extinction of Native Americans in the broader sense as well, although this biological weaponry was not intentional. So this debate caught my attention, as it would be one disease that might have been brought back to Europe as a sort of pyramid's curse. Joe remarked, that's Joe Handelsman, that the paintings of Hieronymus Bosch often depicted noses affected by syphilis, and suggested that could be an argument that syphilis in Europe predated the discovery of the West Indies. However, Bosch died in 1516, with his birth estimated around 1456. His works are not dated. It is probable that his more exuberant works may have come at a later stage in his life, when he might not have needed to adhere so much to social conventions to sell his works after having grown artistically. If syphilis was indeed introduced from the Americas, the grotesque deformations the disease causes may have provided a ghastly inspiration for his marvelous works. As the recording of TWiM-19 lies several years in the past, I wonder if new research has revealed more about the origins of syphilis, that is, whether it came from the Americas or the old continent. I want to thank you all for your continuing effort in making Twix possible. I am sure people around the world have grown accustomed to your voices and expressions, providing them insights, company, and joy. I eagerly read Small Things Considered now, although I must admit that I skip the Talmudic questions as they are way beyond my knowledge level.
Oh, surely not.
Sunny greetings from Nicaragua, where it is now 30 Celsius at 6 p.m. Wow, warm. Kind regards, David. So I did a little hunting, David. And yes, the theory of the origin of syphilis. There are several theories. And in fact, there is one that it originated in Europe, and there's some evidence for that. I think, let's see, where is this paper I found? Probable case of congenital syphilis from pre-Columbrian Austria. This is from an archaeological site. And, they look at the skeleton and they can see changes which are consistent with congenital syphilis. And they say it's probably the first case of congenital syphilis from pre-Columbian Central Europe, so that would indicate that it began there.
Isn't it a problem that syphilis can be confused with a disease called yaws?
Yeah.
Yaws came from the Middle East or the Near East. And I think that's part of the confusion. It can cause symptoms which are similar to syphilis. Mm-hmm. I don't think it's sexually transmitted, though. Is yours? Do any of you know if it's also sexually transmitted? I'm not sure.
I don't think so. No, it is a treponeme, which is the same as syphilis, right? I don't believe it is, because it is typically causing skin lesions, right?
Right.
Direct contact between infected people, non-sexual in nature. Yep.
Yeah, it's not. Yeah,
it's not sexual.
So it's a confusing story, and I haven't followed. But David, I think there is a big literature on it, and I think we should take it up sometime.
Our next note is from Anthony, who sends us an interesting article from Atlas Obscura. It's a website entitled "How a Fake Typhus Epidemic Saved a Polish City from the Nazis." I will leave it to you to read about how this was a faked outbreak and how it helped them out. It's pretty interesting. Did you know about this, Elio? Probably not.
Didn't we talk about this? I thought we talked about it.
We might have, yeah.
So this was an intentional strategy?
Yeah, no, this happened in a town in southeast Poland where two enterprising doctors knew that there was a cross-reactivity, serological cross-reactivity, so that people actually the test for typhus during the war, epidemic typhus, was to do a laboratory test with the serum of patients to see how they reacted to a bacterium called Proteus. Proteus is not an agent of typhus, has nothing to do with it, but for reasons of its own, antibodies against this organism are found in patients with typhus. In other words, that's the diagnostic test. So what they did is they made a vaccine out of Proteus, a harmless vaccine, and inoculated people, and they became positive. and send the serum to the blood to the German labs, and the German labs declared that this was a hotbed of typhus, and they did not go into those towns.
Yep, that's exactly
and
right.
They saved
Proteus.
untold people from having to become slave laborers in Germany, as well as some Jews who were in that town. So this was magnificent. It's quite a story.
Yeah, you're
heroes.
Right. Yeah, that's great. Yep. Thank you, Anthony. Jim writes, I heard this podcast from the Disaster Podcast that's mostly devoted to hospital sanitation. Can you imagine a podcast about hospital sanitation? Who would listen to that? Just kidding. I'm just kidding, because who would listen to microbes or viruses? Lots of people. And this he says, it sure raises my concern about going to a hospital for treatment. I think the emphasis was on disaster cleanup, so the speaker didn't talk about the use of copper, which would be a preventive measure, but certainly one you'd think they'd promote as a sideline. Thought the podcast might be of interest as one that connects the clinical with the research side of things. So the podcast is called the Disaster Podcast, and this is in this particular episode is all about hospitals. So check that out if that's your cup of tea. And Michael would have had a few comments on that, I bet. And one more email from Trudy, who writes, Dear TWiMers, I'm writing this email in response to episode 112. My first comment is regarding the Talmudic question, which I can't find on Small Things Considered for some reason, which generally concerned exceptions to rules in biology. Vincent's comment about how taxonomy is difficult because naming conventions are based on what is currently known reminded me of picornaviruses. When I took my first virology class in graduate school, we learned that the name picoRNA is derived from Spanish, literally meaning small RNA. The name is based on the viral genome, which at the time was considered the smallest known RNA genome. However, since this group of viruses was named, other much smaller RNA viruses have been discovered, such as Rous sarcoma virus, for example. Am I remembering this correctly, Vincent? Anyway, these thoughts can be further supplemented by listening to TWiV 357, which features a discussion about taxonomy and how imperfect this classification scheme can be. I just love how all the sciences complement each other. One can learn a lot by venturing outside one's comfort zone. So I think Rous is not smaller, Trudy, but there are certainly other viruses and viroids that have much smaller genomes. So, well, you're talking about picornaviruses, but yes, other viruses have smaller genomes for sure. Completely unrelated to this discussion, I would like to mention this recent article in Nature, which I found interesting. See links to a Nature article entitled "Page Not Found." Just kidding. Oh wait, I have to copy the whole URL. Let me do that so I can tell you the title of this article. I'm just joking. It's not page not found. Non-coding RNA. Antibiotic tricks a switch. A screen for compounds that block a bacterial biosynthetic pathway has uncovered an antibiotic lead that shuts off pathogen growth by targeting a molecular switch in a regulatory RNA structure. So these are RNAs that are switches, right? Which we've talked about here on TWiM. While most antibiotics function by targeting bacterial DNA, RNA, cell wall, or protein synthesis, this potential antibiotic strategy targets a bacterial regulatory mechanism for synthesizing riboflavin. Apparently, riboflavin facilitates several enzymatic reactions which are essential for bacterial growth and reproduction. As I understand it, this is a previously unexplored strategy for development of new antibiotics. As our imminent antibiotic crisis terrifies me somewhat, I'm wondering if this is something to be excited about. Thanks again for all your entertaining and informative podcasts. They keep me thinking. Kind regards, Trudy. P.S. Having grown up in Romania and having spent summers at my grandmother's house in Transylvania, I have great memories of going mushroom picking with my grandmother, who was very knowledgeable about which ones were safe to eat and which ones weren't. But you're right, nobody in America seems to do this. Americans are mushroom kickers. Right, Elio?
Not quite everybody. There are people who know a lot about mushrooms here, but they're a minority.
Well, Trudy, any new lead on inhibition, and by the way, this targets a riboswitch, which we've talked about on TWiM a long time ago, which is a structure in RNA, which when it binds a metabolite or precursor can turn on a pathway. And that's what they've exploited here, is to mess with this switch. They have inhibitors. I think it's great. And we will see if it works out. if there's any massive resistance, whether it's effective at good doses and so forth, and whether in people it works and gets to the right places and all that.
Specific.
Yeah, and it's specific enough. All right, that will do it for TWiM 116. You can find it at iTunes and also at microbeworld.org/TWiM. And we love getting your questions and comments. Please send them in to twim@twiv.tv. Michele Swanson, who has joined us today, is at the University of Michigan. Thank you, Michele.
Thank you. Go Blue.
Go Blue. How's football, okay?
Yeah, we've had some exciting last-minute contests, but we've got a big match with Penn State in Happy Valley this weekend. So,
good luck.
Thanks.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure, as always.
Always good to have everyone. And I'm Vincent Racaniello. You can find me at virology.ws. I would like to thank the American Society for Microbiology for their support of TWiM, and Chris Condayan and Ray Ortega, who work for ASM, for their technical help with the podcast. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.